আছে কিনা? If you have no question on previous lectures, I would like to start uh, my lecture on secondary metabolites. Some of the students may attended the course Bioactive Natural Products, but here Hussain Sorowardi is a PhD student. He has no scope to attend that course. So bioactive secondary metabolites, fundamentals and the diversity is very important to get better understanding on ecological chemistry. This is why I am giving you the fundamental understanding on secondary metabolites. Let us start the course. Yes, secondary metabolism in plants. Although I mentioned in plants, but we can discuss, elaborately, because secondary metabolism is not only present in the plants; it is robustly present in microorganisms, insects, and other organisms also. Metabolism, possibly, the history of metabolism and what is metabolism. Metabolism, it is derived from a Greek word metabolismos. The meaning is out through, is the set of chemical reactions that happens in living organisms to maintain life. I am sure in your biochemistry, undergrad biochemistry or master's level, you receive the training on different kind of metabolism, how energy is generated in the form of ATP from the glucose, or how tricarboxylic acid cycle or Krebs cycle is going on in the mitochondrial respiration to release the energy from the different carbon containing compounds. So lots of biochemical processes are going on in the cell, in all the cells, not only plant, but also all organisms. Metabolism can be broadly divided into two groups. One is called catabolism. Catabolism means breaks down organic matter. For example, harvest energy in cellular respiration. For example, acetyl CoA enters into the TCA cycle and release lots of energy in the form of ATP, and the breakdown product is carbon dioxide and water. So glucose breakdown, like glucose, glycolysis, the process of generation of energy from the glucose or like that is called catabolism, that is breakdown of organic matter. Anabolism uses energy to construct components of cells, such as protein, nucleic acids. That means, in the cell, structural or functional biomolecules, big molecules like protein. Protein is structural molecules in the cell, as well as nucleic acids like DNA, RNA, is synthesized from the small molecules using the energy in the cell, and it is called anabolism. That means small molecules, used to synthesize the big molecules. And in case of catabolism, big molecules break down into the energy. So these two groups of metabolism is going on in the cellular system all the time. Santorio, in 1614, in his book Ars de Statica Medicina, published experimental results in human metabolism. So human metabolism, recorded history tells us that people know about the metabolism since early 17th century. Secondary metabolism. Metabolism can be divided into primary metabolism and secondary metabolism. Today we shall discuss on secondary metabolism. Metabolites. Uh, Secondary metabolism is associated with the synthesis as well as function of the uh, secondary metabolites. What are secondary metabolites? In the journal Science in 1959, defined secondary metabolites are organic compounds that are not directly involved in normal growth, development, and reproduction of an organism. That means they are associated with other purposes, not directly involved in growth or development or reproduction of an organism, but they are very, very important. I shall discuss later on. And in ecological chemistry, they are vital. Secondary metabolism, the process of biosynthesis of secondary metabolites as well as their functional aspects is the subject matter of secondary metabolism. Primary metabolism, that means synthesis of primary metabolites. What are primary metabolites? Primary metabolite is a kind of metabolite that is directly involved in normal growth, development, and reproduction. If I ask you, cite an example, for example, a protein. Protein is a primary metabolite. An amino acid among the 20 essential amino acid is a primary metabolite. A molecule of glucose is a primary metabolite because they are involved in growth, development, and reproduction of the organism. But if I ask you what are secondary metabolites, any example? For example, in the plants, alkaloids are secondary metabolites. Even flower colors, flower aroma, all are secondary metabolites. They are not directly involved in growth, development, and reproduction of the plant. Metabolites, what are metabolites? This is a general, question, because now primary metabolites and secondary metabolites. But if I ask you, what are metabolites? Metabolites are intermediates and products of metabolism. Metabolism is a process by which different molecules are synthesized, or in the big molecules like nucleic acids or protein, or breakdown of the organic compounds to generate the energy or to become the small molecules. So metabolism is a multi-step process which is triggered by the enzyme. Enzyme is has a catabolic function for each and every step of the metabolism. An enzyme, specific enzyme, is involved. Sometimes some cofactor also involved in the process. So metabolism is the set of chemical reactions that happens in living organism to maintain life. So now it is complete. Metabolism, metabolites, primary metabolites, secondary metabolites. So the basic concept, I think it is clear, because these topics are included in biochemistry at undergrad as well as master's level. How secondary metabolites are synthesized in the plant body. How primary metabolites are synthesized. In case of plant, primary metabolites are synthesized basically through the process of photosynthesis, because in the photosynthetic process, photon, the natural energy source coming from the sunlight, is used to split the molecule of water, and as a result, water molecule gets electron, one higher level of electron, and it releases it, and that electron actually pass through the electron transport chain and produce the ATP energy, and that energy later stays in the photosynthesis, used to fix the carbon dioxide into the plant cell. So photosynthesis basically two phases. One is light reaction, another is carbon synthesis or dark reaction. Dark reaction, actually carbon dioxide fixation is taken place, and light reaction, actually photon is used by a chlorophyll and photosystem to get the energy first in electron form, and that electron is through electron transport chain in the plant generate ATP and NADPH2, and they are the energy source required for fixation of carbon into the plant cell to produce glyceraldehyde 3-phosphate, which is the primary outcome of the photosynthesis. And glyceraldehyde 3-phosphate actually used to synthesize glucose and many other amino acids, all other organic compounds in the cell. So primary metabolism, you know how it is happening, and glucose is the central part. From the glycolysis, phosphoenol pyruvate carboxylase is produced in the glycolytic process, and pyruvate actually enters into as acetyl-CoA in the TCA cycle. But in the TCL cycle, acetyl-CoA, some of the acetyl-CoA can be utilized in the mevalonic acid pathway, or melonyl-CoA can be converted into melonyl-CoA, and they are used to synthesis of polyketides as well as isoprenoids or complex isoprenoids. Isoprenoids and complex isoprenoids, they are terpenoid compound. Similarly, pyruvate can be converted into aliphatic amino acids, and amino acids are the precursor of alkaloids, because alkaloids are derived from the amino acids. So most of the amino acids, 20 essential amino acids, are used for synthesis of protein, but some of the amino acids, some of the acetyl-CoA, they are used to synthesize the secondary metabolites. So alkaloids are secondary metabolites. There are various kinds of alkaloids. Similarly, aromatic amino acids also generate in the cell through the pentose phosphate pathway, which result erythrose 4-phosphate, and then erythrose 4-phosphate converts into shikimic acid. And shikimic acid, there is a pathway in the cell, and lots of enzymes are involved there. So from the shikimic acid pathway, aromatic amino acids are produced, and they are used. Some of the aromatic amino acids are used in the phenylpropanoid pathway to synthesize different kind of flavonoids and complex polyflavonoids, or phenolic compounds, which are secondary metabolites. So hydroxybenzoic acids can be generated from the shikimic acid pathway in another chain, and this molecule also be used in phenylpropanoid or flavonoid or complex flavonoid or phenolic compounds. So what is the summary of this discussion? Summary of this discussion is that secondary metabolites are derived from the primary metabolites, but they are not involved in growth, development, and reproduction of the organisms. Now you may ask question, why a plant or other organisms use lots of metabolic pathway and use lots of energies to synthesize these secondary metabolites? There are lots of reasons. One of the important reason is the fitness of the organism. For example, flowers synthesize different anthocyanins or different medicinal compounds, toxic compounds, antimicrobial compounds to protect themselves from the attack of pathogen. Similarly, microorganisms also do the same. But you may ask a question, why human? What about human? Human actually do not need to synthesize secondary metabolites because we have powerful immune system, antibody and T cell and many other mechanisms to fight against the pathogen. But in case of plants, in case of microorganisms, they have no such, powerful defense or immune system. So their immune system is different and fascinating, and secondary metabolites are involved there. Among the secondary metabolites, there are several groups. We can primarily divide into them into terpenes. Terpenes, the largest group, and then alkaloids. Terpenes are derived from the C5 precursor isopentenyl diphosphate IPP synthesized through the from the acetyl CoA in the metabolic pathway. Uh, Scientists already discovered the biosynthetic pathway of different kinds of terpenoidal compound. Second group is alkaloids. Alkaloids are very famous. Even in the ancient time, people have been using plant alkaloids. Alkaloids contain nitrogen because they are derived from amino acids and they are very powerful compound. For example, nicotine, caffeine, morphine, or cocaine, very heroin, all the compounds, they are alkaloids. This is why from the ancient time, people use alkaloids as drug as well as medicine. There are phenolic compounds very important for the life of the producer as well as life of other organisms, especially in human, because the more phenolic compounds you can take from the plant sources, the more you can protect from the diseases. So phenolic compounds, they can detoxify the superoxide generated in the body. So phenolic compounds are very, very important, and they are produced through the shikimic acid pathway or malonate or acetate pathway. Uh, Secondary metabolites can also be classified into different ways. For example, nitrogen containing secondary metabolites or non-nitrogen containing secondary metabolites. Nitrogen containing secondary metabolites are alkaloids, non-protein amino acids. Only 20 essential amino acids are involved in synthesis of protein, but there are lots of non-protein amino acids discovered in plants and other organisms. There are some amines, cyanogenic glycosides, glucosinolates, all contain, nitrogen in their structure. So nitrogen containing secondary metabolites, and without nitrogen, non-nitrogen containing secondary metabolites are terpenoids and phenolic compounds. Terpenoids can be divided into monoterpenoid, sesquiterpenoid, or diterpenoids, triterpenoids, polyterpenoids. Steroids also, terpenoidal compound. There are some saponins, beta test of the compound, for example, cucurbitacin, or like that in cucumber. Sometimes cucumber is bitter, or the joint head of the cucumber fruit, you can find beta test. They are saponins. So phenolic compounds can be classified into flavonoids, polyacetylens, polyketides, and phenylpropanoids. So there are lots of these numbers. I am giving you the approximate, and every day new secondary metabolites are discovered, and they are published in the science. And most interesting compound discovered for the first time for human use was the penicillin. Penicillin was discovered actually in 1932 or 34 by Alexander Fleming from a fungus, Penicillium notatum, and it was the first antibiotic commercially being used and saved life. And after that, thousands of antibiotics have been discovered from the plant sources as well as microbial sources, and all those are basically secondary metabolites. So compartmentation of secondary metabolites biosynthesis in the plant cell, for example, mostly in the cytosol. Hydrophilic compounds are found in the cytosol. So in the cell, where these toxic or as well as functional metabolites are stored, this means the compartmentation. So chloroplast alkaloids like caffeine or terpenoids, they are stored in the chloroplast. Chloroplast is an organelle, cytosol is an organelle, mitochondria, some amines and alkaloids, vesicles, alkaloids, or endoplasmic reticulum like hydroxylation, step or lipophilic compounds. So a plant or any other organism, they have the mechanism to store those molecules in certain, organelles in the cell. For example, mint leaf, when you touch the mint leaf, you can get the strong aroma because mint leaf under microscope, you can see they contain the bulbous structure called glandular trichomes, hairy structure. So if you touch, immediately they burst and release the terpenoidal compound. They are basically antimicrobial and to ward off the herbivores so that they cannot eat the mint. But we love those, compound and menthol. And menthol has antimicrobial properties, but a small amount we can easily eat, and it is good for burhani and other dessert we can or salad we can use. Here you can see in the mint leaf, leaf surface, here is the microscopic structure. You see a ball-like structure. This is highly magnified, and it is filled in, terpenoidal compound, volatile compound. So when you touch, immediately they burst, and this is the mechanism of chemical, defense of the plant. And here you can see thyme glandular trichomes. So these are the glandular trichomes under scanning electron microscope, and they are filled in the chemical substances. Lemon leaf, if you crush a lemon leaf, they contain this type of secretory cavity full of different kind of aromatic volatile terpenoidal compounds, so you can get strong aroma. And pine resin duct, pine tree, if you cut, you can see milking substance are coming out, or rubber tree, if you cut, you can see milking substance are coming out. They are basically the defense molecules, and they are secondary metabolites. So there are lots of examples, and this is the last slide for today. Function of secondary metabolites. Why organisms, plants and other organisms, they produce diverse kinds of secondary metabolites. In case of plants, let us consider an example like plant, because plants are sitting duck, they cannot move away, and to defend themselves from the attack of herbivore, including human, they need some chemical weapons. So plant secondary metabolites are basically mixtures, not a single compound, and variation in time, space and development stage. So different time, different developmental stage, the mixture ratio of different compounds are hugely varied. So what they do, plants, always open to the sunlight. This is why they need some sunscreen-like substances, UV protection substances. So some flavonoidal compound, they can protect the plant cell from the UV light coming through the sunlight. So this is very interesting, and scientists discovered many compounds from the plants which we are using now as sunscreen. When you go to the beach or sunlight, sportsmen, you can see they use different cream-like substances which contain the flavonoidal UV protecting substances. Interestingly, when I was in Japan, I discovered 5 new dihydroflavonols from a wild plant called Caphila or Giga or Lania coromandelica in scientific name, and they are now Canadian company are now using in the sunscreen. Giga tree, wild tree, people use as a hash, and their bark is very thick, and the bark contains the UV protectant dihydroflavonols. And those dihydroflavonols now we can use as our sunscreen. So they use smart system to protect themselves from the attack of UV. But human has no such system. This is why you need to use sunglass. You need to use umbrella to protect yourself from the UV, because UV is damaging, can cause lesion in your DNA, and even can result cancer. But our DNA has a repair and maintenance system, so most of the damage caused by the UV are repaired, but if remains, then it can result cancer. Uh, the secondary metabolites are also used as attractant or attraction. For example, pollinating insects are attracted by the aroma or a scent. They are basically terpenoidal compound, or the color. Seed dispersing animals, because fruits, when it is ripen, you can see aroma is coming out, jackfruit or any other fruit. So birds, they eat, and they or other animals with the seed, they eat, and they when they move other places, they also carry the seeds and disperse the seeds. Root nodule bacteria, some leguminous plants, that means bean plants, they have the mechanism to attract rhizobium or bradyrhizobium bacterium to allow them to make colony in their root called root nodule, and they can fix the nitrogen and offer the plant. So secondary metabolites in plant, they are very important for bacteria to recognize which one is their host, legume plants. Induced volatile attractants, plants even sometimes use volatile attractant compound, even when they are attacked by the insect. Immediately they synthesize volatile compounds to tell other plants to activate their defense system. That means plants can use even Wi-Fi and internet system. That means wireless. They use the volatile compounds to talk with each other and to warn others, so that they can make enough defense system to protect the enemy. Predatory organisms, there are some insect or organisms which are not harmful for the plants but harmful for their enemies, like a ladybird beetle. Ladybird beetle can eat the grub of other insects, egg of other insects. So they attract the predatory organism, their friends, so that they can get the shelter in the plant, and in return they help plants to protect themselves from the attack of the enemy, that is other insects. They eat the eggs as well as larvae of other insects. Okay, and the defense, you see, defense is number one function of secondary metabolites produced by any organisms. So one of the important defense for against the herbivores, for example, in human, we have more than 400,000 plants in this earth, but we have selected 15 to 20 species as our food crop, but all other plants are outside of our food crop. Do you know why? Because they contains different toxic compounds, secondary metabolites, which we cannot digest or tolerate. So similarly, in case of insects and vertebrates, so they have the limited choice. For example, an insect which is toxic, which can infest the rice, never infest the jute, because jute secondary metabolites are deleterious to that insect. This is why host a range of the herbivore is limited by the secondary metabolites, so it is a defense mechanism. Some defense compounds are repellent or deterrent, and some are toxic. Even if we take some of the plant, like Socrates was killed by the alkaloid. But do you know what was the alkaloid was used?
Ha- Hemlock.
Hemlock, sir.
Hemlock. So hemlock is an alkaloid, was used in ancient Greek people to kill the prisoner. So hemlock is also a defense molecule. Similarly, defense molecule plant use to protect themselves from the microbial attack, like bacteria, fungi, and virus. Some of them are growth inhibitory, some of them are toxic, some of them are just disturb the physiology of the pathogen. And plant also use secondary metabolites in competition with other plants, which are called allelopathy. That means plant use some phytotoxic compound, not allowed to grow other plants nearby, or even stop the germination or seedling growth of the other plants. So lots of strategies are used by the secondary metabolites produced by the plants. Similar case happened in case of microorganisms also, because microorganisms are reservoir of incredibly diverse secondary metabolites. For example, streptomyces species. One scientist got Nobel Prize from Japan. He in his whole life he studied secondary metabolites from the streptomyces. Streptomycin, which was a very famous antibiotic, and lots of antibiotics have been discovered from the streptomyces species. And streptomyces, a soil-borne bacteria abundant in the environment, but very few studies have been carried out streptomyces present in our country. Satoshi Omura in Japan, he got the Nobel Prize because he discovered 400 new secondary metabolites, antibiotics from the genus Streptomyces. And many of them are in the practical use as a pharmaceutical drug, including, ivermectin or streptomycin, and many more. So this is all about today's talk. I think it is a stimulating talk for you, and we shall discuss much more. So here you can see how, by using small molecules, plant which are standstill, they can do they can communicate and interact with the other members, including human, in the ecosystem. And this is why you see all of the organisms on this earth, directly or indirectly, depend on plant for the energy source. But even though the earth is green, because of the production of secondary metabolites, otherwise you cannot find any plants in this earth, because all the animals, humans, and other organisms, they get energy from the plants. So plants will be extinct if there is no such defense mechanism. So this is all for today's lecture, and tomorrow I shall go to the campus. So it would be very nice if we can set time evening, or otherwise we can set time on Saturday or Friday for the makeup class, because tomorrow we have lots of meeting, as well as our laboratory renovation process is going on, with a fund from the Krishi Gaveshana Foundation. This project virtually completed, but we have received clearance for a few months extension. So I need to utilize all the money to furnish the laboratories, and really the job is about to end. So thank you so much for listening this talk, and I hope you can ask some question. So unmute yourself and ask the question. Ruhul Amin?
Yes, sir. You have mentioned that the secondary metabolites produced from the plant, they may be, whether they cannot, they will not be lethal, but they may produce the toxic effect. So sunscreen, it protect us. So I have my query is that when they goes for industrialization, so how they mitigate the toxic effect in this secondary metabolites.
Industrialization of any plant product.
Yes, sir. Yes.
Actually, human over the years of evolutionary process, they adapted to the toxic effects of some of the crops. For example, 20 species. I told you that all over the world we use 15 to 20 species as our food crop. They also produce secondary metabolites, those plants, but we are adapted to those toxins because over the years of evolutionary process, our digestive system has some enzymes to break down those toxins. But a most important point here is that any food crop or any food item never take too much, because then you get too much toxin. Plant has ability, a human has ability to detoxify or tolerate certain level of toxin with the nutritious food. So if you take too much, then it would be problematic for you, and you may face the toxicity. And too much of any nutritious food, even not good. Obesity is really a big problem in many rich countries like USA, and obesity is due to the over nutrition, or getting the nutrition, especially carbohydrate and other nutrition very ready format, and which are not metabolized properly for the fitness of the human. So this is why they become so fat, and their immunity also decreased. In the industrial process, when always people always consider the anti-nutritional and other compounds. In some cases, there is a mechanism to separate the anti-nutritional substances. For example, how to say, cotton seed oil is good for cooking and eating, but it contains gossypol. Gossypol is a toxic substance for human. Like mustard oil contains the erucic acid, not good for our health. If you take too much erucic acid, it would be carcinogenic even. And gossypol also similar. So scientists are using several strategies to separate the gossypol or erucic acid. For example, in Canada, they have cannula. Cannula, do you know what does it mean? Same brassica plants, that is oil yielding mustard plant, which has no ability to synthesize the erucic acid. That means they silence the gene by breeding or some ways. Similarly, gossypol free, mutated cotton plant already developed by biotechnology. So what you can do, you can silence the gene, or many other ways you can tackle those problems.
Thank you.
You are welcome. Any other question?
Sir, colchicine is also secondary metabolites.
Yes,
Uh,
colchicine is an alkaloid, and it is a carcinogenic compound. If you apply colchicine as a very low dose, and it is found in the root of a plant, colchicum. So if you use, it can cause the doubling of chromosome number in the cell. Really very dangerous compound for human. If human exposed to that compound, it is terrible. But scientists discovered its principle. Now it is used in the breeding of double haploid or like that. So double haploid technology we can easily deploy it to tetraploid we can get by using the colchicine. Yes, it is a secondary metabolite. There are many peptides, are also secondary metabolites. For example, wasp venom also, they are peptides. Even snake venom, some of them are peptides. So very toxic for the human. They are not used as growth and development of the organism, but they are used directly as a defense, that means chemical weapon of the producer. And discovery of those toxins are very important because, for example, morphine. Morphine is, illegal to trade, but if no painkiller, work in some cases, morphine is suggested as an injection. And in case of morphine, the big problem is once it is injected to a person, within a few days, people cannot survive without that morphine, because addiction is associated with the practice. So 2 or 3 times, 3 doses if someone takes, and then it must continue. I met one Bengali scholar in Bergen, in Norway. He had a serious car accident, and his spinal cord was damaged. So he has to, receive morphine every day, 8 hours interval. So after 8 hours, without any clock, he can tell that 8 hours is over. And like insulin, he himself takes the morphine. Without morphine, other painkillers didn't work on him. Any question? If no
Sir,
question, then thank you so much. And next class, I shall discuss on an issue. How can you, uh, do some assignment? And in case of assignment, I shall give you some guidelines how to write a review, because this is a fascinating topic. If you write a review, maybe we can publish in the journal as well as a book chapter. So as you are small in number, 5 persons, so I can guide and supervise everyone so that everyone can write a review article on a particular topic. For example, pheromones in agriculture, like this, or eco-friendly agriculture. So there are lots of topics I can propose uh, that could be published, and you can write a review, and that review even you can later use as your seminar paper in the because you have to attend a seminar course. So double or triple benefit so that you can get, and this would be a good motivation for you to learn the topic very deeply and with high enthusiasm. What do you think this idea?
This is a very great idea, because it will enrich our subject knowledge, and to critically thinking the ongoing research, and ongoing research gap and future perspective.
Yeah, this is a very nice comment. Yeah, I had one student only in a course, nanobiotechnology, and he wrote a review article, and it is now under review of a journal, Current Research in Biotechnology. I am optimistic that it will be published as a review article in an international journal published by the LCBR. So you can imagine from a class test, uh, people are, if a student get such a reward, like pollinators are getting the reward as an ecological chemist, I should always consider how you can get much more reward from your work, from your learning, and how much you can enjoy. And without enjoyment, learning would not be stable. You can get a or a plus, doesn't matter, and finally you will forget everything. But I hope you will enjoy this course, because this course is unique, and I believe in the past you didn't embrace such a different, fascinating subject, right?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, then thank you so much.
Assalamu alaikum.
Walaikum salam.